Okay, good morning everybody and welcome to this Eucleg training workshop. I'm just here to help things run smoothly and to introduce the speakers, and I will be asking the questions that you put. If you have questions, we'd be very grateful if you could put them into the questions tab, which is on the right hand side of your screen. There is a chat function which is there for you to just say hello to other people who are on the call today. And the other thing I'd like to mention before we start the official presentations is that for those of you who haven't yet registered for tomorrow, it is not too late, but because it is necessary to register for the two sessions separately. So if you have not done so, please register for tomorrow if you're interested in hearing the excellent range of talks that we have then. So just to I should just ask Juliet now to sorry.
Well,
I got the wrong presentation there.
Yeah, I think there is an introduction by David Lloyd first.
Yeah. No, it's a no. Sorry, no. The introduction is the introduction will be from. I've got to find. Here we are. So Juliet, if you'd like to, sorry, Bernadette, if you would like to introduce the project, please.
Okay, good. Hello, everybody. So I'm really pleased you can attend this workshop or this training. I'm Bernadette Julier. I'm from France. I'm working at INRAE, and I'm the coordinator of this project, Eucleg. So Eucleg, the title of this project is Breeding Forage and Grain Legumes to Increase EU's and China's Protein Self-Sufficiency. In fact, this project has been launched after, this project has been prepared after a call launched by European Union in 2016. And the title of this call was a joint plant breeding program to decrease the EU's and China's dependency on protein. And the call included many very important words: forage, animal feed, proteins, productivity, climate change, diversification, and so on and so on. And also call for collaboration with Chinese colleagues. What's the problem with proteins in Europe and China? Protein dependency of Europe is about 69%, and China imports 60% of soybean world market trade. And most of the soybean is imported from the USA and Brazil, or South America. And the tendency is quite different in Europe and in China. In Europe, the imports that you can see in the section other countries mostly is more or less stable, but important. And in China, the imports are recent. They begin about 20 years ago to feed the animals, and the tendency is towards an increase of imports. Okay. And seen from the economic point of view, in fact, proteins are the section of plants that are largely imported in the European Union, and it's a problem, an economic problem compared to other crops. Well, what about the proteins? In fact, the question here is to transform nitrogen or dinitrogen, that is a gas, into proteins. So dinitrogen is a very stable molecule, and it's 68% of the atmosphere. Probably most of you know everything, know this very well. But so dinitrogen is a molecule composed by 2 atoms of nitrogen bonds with 3 very strong covalent bonds. And nitrogen is a component of proteins, and proteins are vital molecules. There are two ways to transform dinitrogen into reactive nitrogen. First one is a chemical synthesis that is carried out in industries. And by using large quantities of fossil energy, and in the process done by Aber and Bosch engineers, the nitrogen is transferred into ammonia. The other way is to use the symbiosis between legumes and bacteria, soil bacteria called rhizobium, and that are able to transform, to do the same reaction, in fact, with the help of enzyme, the nitrogenase. And then the plants are able to assimilate, absorb the ammonia and transform it into amino acids and then in plant proteins. We also have a problem with the nitrogen cycle at the world level. Nitrogen enters in a plant compartment, mostly from nitrogen fertilizers that enters into croplands and also grasslands. The second way of entrance of nitrogen is from nitrogen fixation with legumes. And the third and very small part is deposition from the atmosphere. So the plant grows, there is some leaching, and nitrogen goes to the ecosystem. The plants are eaten either by livestock or human population, of course. And again, there are some waste from livestock or from humans. And at the end, it's a completely open nutrient cycles with huge losses that cause pollution. So there is a question for environmental sustainability. And we also see here the part of nitrogen fertilization into the entrance of nitrogen in the crop production, and a very small part of nitrogen coming from fixation at the end. So the global statement is that we need to expand plant protein production, and we need to increase nitrogen fixation, and as a summary, to grow more legumes and to produce more proteins from legumes, to rely on this equation of nitrogen fixation. So in Eucleg, we have focused on the 5 major species in Europe. So these legume species are able to produce protein-rich products while providing positive environmental footprints. What are these products? These products are either forage or grain. So we have studied alfalfa or lucerne and red clover. And as grain legumes, we have worked on pea, faba bean, and soybean. And the Chinese colleagues have worked on alfalfa and soybean. Only they are the most important species in China. So forage are used to feed ruminants, mostly, a little bit monogastrics, and very rarely human. While grain legumes are used for monogastric and human food, and partly for ruminant feed. The expected impacts of Eucleg is to increase protein production where legumes are really grown. I mean, in the regions where legumes are already grown, but also to increase adaptation of legumes to more pedoclimatic regions, so we are able to grow legumes where we are not able at the moment. So at the end, we need to produce feed and food, and we have to achieve a high yield and improve yield stability. And this can be achieved through many improvements related to general adaptation, resistance to biotic and abiotic stresses, adaptation to climate change, Also, the quality seen depending on the species as a protein content, protein quality, or forage quality, and also a decrease in anti-nutritional compounds. So Eucleg use genetics as a lever to achieve these goals. Eucleg is based on the use of genetic resources, but also elite material coming worldwide, even if material is mostly based from Europe and China. The idea is to have a large phenotypic evaluation on many traits. The traits have been established by species experts who are able to see what are the most important traits. We have developed molecular tools to genotype the accessions. And we have studied diversity, the genetic determinants of traits, look for associated markers, so markers associated to the trait variation, and worked on the potential of genomic selection to improve the breeding programs. So the outputs will be, or the impacts will be for breeding and for the release of new varieties in the future. So what are the objectives of Eucleg? We had objectives at different levels, of course. Some objectives at the scientific level. First, it was to broaden the genetic base of legume crops, because usually breeders use their own genetic pools, but it's sometimes important to go outside of this pool, and to analyze the genetic diversity of European and Chinese legume accessions using, of course, phenotypic traits and also molecular markers. We have the objective to analyze the genetic architecture of key breeding traits using GWAS, and also evaluate the benefits brought by genomic selection to create new legume varieties. At the technological level, we have developed searchable databases containing passport data as well as agronomic and genetic features, and also developed molecular tools and data that will be available for future programs. At the applied level, that is breeding in this case, the objectives are to develop tools for genotyping and also implement data management and analysis that is quite compulsory in genetic programs and genetic breeding programs. Just here to show you the Eucleg workflow that will be the basis of the presentations of this workshop. So we have the 5 species, and we have adopted more or less the same scheme for all the species. Based on the use of genetic resources, for which we have to improve access and information. So at the left-hand side of my screen, we have collected between 200 and 400 accessions. We have developed or make use of a genotyping platform depending on the species, and the objective was to obtain more than 10,000 markers. We have genotyped accessions with these genotyping platforms. We have also phenotyped the accessions in different conditions, field conditions with multi-local trials, but also control conditions, especially for diseases or drought and some traits that are more easily under control conditions. All these results have been transferred to databases handled with the help of this program, Progeno. And then we are analyzing these results to detect genetic diversity, genetic structure among accessions, study genotype cross-environment interaction, and also carrier with GWAS and genomic selection. Okay, so the objectives of this workshop is to disseminate the results obtained so far, so progress on grain legumes that are presented today, and progress on forage legumes. That is a program of tomorrow. We have the wish also to share general considerations about the design of multi-location experiments, about genotyping methodology, about plant genetic resources. And the idea is to talk and discuss with you, with legume breeders, with scientists, to imagine future breeding of legumes, what I could call post-Eucleg breeding. So you have the program in your hands, so it's just here a copy. And I would just, as Catherine explained, it is still time to register for tomorrow's session. Some of you are registered for today's session only, so it's still time to register if you follow the link. Okay, so I think the introduction is finished, so we can continue with the program. I wish you a very pleasant workshop.
Thank you very much, Bernadette. That was an excellent introduction. We haven't had any questions on that. Just to remind people, I expect that there will be more questions on the main talks that we have later on. But just to remind people, if they wish to ask a question, if they could write it in the questions tab, please.